Welcome, everyone. So this is the webinar series jointly hosted by COPSS and NISS for the topic of leadership in the intersection of statistics and genomics. I'm Jessica Lee. I'm a professor in the Department of Statistics and Data Science at UCLA. I'm also a member of the 2023 class of the COPSS Leadership Academy. Just as a little background, COPSS is the Committee of Presidents of Statistical Societies. It is made up of various statistical societies, including the ASA, ENR, WINNER, IMS, and the Statistical Society of Canada. And COPSS bestows many awards each year, including the new COPSS Leadership Academy, which is recently renamed to be the COPSS Emerging Leader Award. So the purpose of this award is to recognize early career statistical scientists who show evidence of and potential for leadership. It also provides a mechanism for us to share our vision of leadership with the field. From this emerges the webinar series, which is co-organized by COPSS and our partner at the National Institute of Statistical Sciences, short as NISS or NISS. These webinars are an opportunity to feature leaders throughout the discipline, invite them to tell their leadership stories, and share their insights. This is the opportunity for today is about the intersection of statistics and genomics. In our profession, many of us think about the impact statisticians can play in scientific research. Genomics is a prosperous field where rapid technology development led to many data analytical challenges. Hence, genomics is an important application field of statistics. However, for statisticians diving into genomics, figuring out how to take charge and lead the way is still a big question mark for many of us. So today we're fortunate to have two leaders who have successfully straddled the line between statistics and genomics. Our two panelists are Dr. Rafael Irizarry and Dr. Mingyao. So Dr. Rafael Irizarry is a professor and chair of data science at Dana-Farber Cancer Institute and Harvard School of Public Health. He's a renowned statistician with a specialization in genomics. He's known for open source statistical methodology shared through Bioconductor in R. Rafa has mentored numerous trainees and offers popular online courses in data analysis. He was a recipient of many prestigious awards, such as the COPSS President's Award and Benjamin Franklin Award. His work impacts fields ranging from infectious diseases to COVID-19 vaccine effectiveness. Dr. Mingyao is a professor of biostatistics at University of Pennsylvania. Her research spans statistical genetics, genomics, bioinformatics, and computational biology. Her recent work focuses on understanding cellular heterogeneity in human tissues using single-cell and spatial transcriptomics data. Dr. Mingyao has received notable accolades, including the International Statistical Institute member in 2014, and she became Fellow of the American Statistical Association, ASA, in 2018, and recently she was named a Fellow of the American Association for the Advancement of Science, AAAS, in 2021. The structure of today's event will be as follows. First, we will have each panelist give us a 10-minute talk about their leadership journey. Then I have prepared some questions to ask. Finally, we'll open up the Q&A to our audience. So first, we will thank our two panelists to share their journey with us. So maybe Dr. Rafael Irizarry, you can start first with your leadership journey. Thank you.
My leadership journey. So I got my PhD from stats in Berkeley, and I began as an assistant professor after that in Johns Hopkins Biostat department. That's where I did not come in knowing being an expert in genetics or anything like that. Genomics wasn't really a field that was that popular in statistics yet, but they were starting to come. This isn't like the late 90s. They were starting to more and more biologists were generating data and using these new technologies, and they eventually came to biostat departments where the chair of the department at that time, Scott Seeger, connected them with me, and that very quickly became my main area of expertise. Then in terms of leadership, in 2013, I was recruited to Harvard and Dana-Farber, and about 5 years later, they convinced me to become chair of the department, and I've been doing it since 2018. And during that time, my main goals have been the one that all chairs have, which is to bring the best people possible to the department to move forward the discipline. But also, in my case, I'm in a cancer center where we have hundreds, thousands actually of faculty, many of them generating data, relying on data. So part of my mission is to make sure that the best data analysis happens in the institute as a whole. So that's also a big part of my role here, is to try to provide support for others in the institute through collaborations and service.
Thank you very much, Rafa. So maybe I can follow up about your journey. So after you became the chair of the department, or once you joined Harvard, so how would you see the differences for a statistician working in a biostatistics department alone versus working in a cancer center? So what are the unique opportunities and challenges in a cancer center?
So I mean, I don't know how much this extrapolates to all cancer centers, but it's probably true in the big ones that most of you know, like MD Anderson and MSK, Moffitt, these places, and then most universities have a cancer center within their medical school. So what are the challenges are that the main challenge for faculty in our department is that and I think it's true in most, but again, I don't want to generalize too much. It's not always the same in every place, but we're mostly soft money. And that means that you get hired, the way it works is you get hired with a very large startup. You get, funds to start your research going, but you get that with the assumption that you're going to try to get your own funding in 3 to 5 years and start bringing in grants to support your research program. That's in contrast, hard money positions, which are not necessarily all biostat departments. In fact, I think most aren't completely hard. But in stat departments in schools of arts and sciences, they are usually hard money, and there you get 9 months of your salary covered for teaching. So the startups that aren't nearly as big, although there are exceptions, but you don't rely on grants to cover your salary, but you have to teach for that to happen. Again, there's exceptions, and people sometimes negotiate very little teaching and large startups and also hard money, but I wouldn't say that's the norm. So that's the big difference. Now, biostat departments are in between, depending which one. Some have about half hard money, some have almost none, and then there's in between. But so the advantages of those two and disadvantages, I would say the advantage of the soft money institutions are, again, I'm generalizing. One is that you don't have to teach as much, so you have more time for research. The salaries tend to be higher, again, in general. Those are the two main advantages. And then the other one, at least for people who are apply like me, is that you get direct access to a lot of interesting statistical challenges, in particular in genomics, but there's also clinical trials and there's correlative studies. There's imaging, there's a lot of things happening. And one thing that happens in science in general that I see in biomedical sciences is that cancer research, because it gets a lot of funding, and it's also something that as a society we dedicate a lot of resources to, and think it's very important to cure the disease that most kills younger people. That there's a lot of the biologists in the forefront of genomics are often in cancer centers. So they're generating new data sets with new technologies. They're trying, everybody's trying new things, whatever they think that could help. So then you have colleagues that are working on problems that are generating new data sets. So you get to be first in a way in terms of having access to that. Again, you can have a collaboration with a cancer center scientist outside of not being a faculty in a cancer center, but we have closer connections. And of course, basic biologists also do a lot of that too, right? But I would say that in general, a lot of the newer technologies are very commonly available and in need of collaborators in cancer centers. The disadvantages is the fact that you rely on grants. There's more stress because it's not guaranteed. You have to constantly be applying and thinking about how you're going to fund yourself. So that's of course more stressful. And it could be in a place where you don't have support and you have to cover like all your salary. It can be quite stressful. And that does takes away time that you can spend on thinking, on actually deep thoughts and really looking at the data when you have to worry about funding. So I would say that's the main disadvantage compared to a hard money position. Now you asked about biostats can be soft money. So the same arguments apply, and then in biostat departments, you do also have access to a lot of interesting problems in public health, environmental sciences, and epidemiology. There's a lot of data rich disciplines there as well. Does that answer your question?
Yeah, thank you, Rafa. I think we have some similar questions at the Q&A. So thank you for the introduction. So now we'll let Dr. Mingyao Li to introduce her leadership journey. Thank you, Mingyao. Sorry, Mingyao, you are still muted.
Okay, so I prepared my slides. Do you see it? Yeah, great. So I don't really have the leadership experience to share, but I can share my journey in statistical genetics and genomics. So I prepared one single slide to summarize my career and the research path so far. As you can see that I got my PhD a long time ago. I did my PhD in biostatistics from the University of Michigan between 2000 and 2005. And I worked with Mike Banke and Gonzalo Abcasis for my dissertation work. So during my PhD study, I learned something that is very traditional in statistical genetics. I learned linkage analysis, and I developed methods to analyze data for linkage analysis. And I think many of the people in the audience may have not had experience of working in this area, but that is how I started my research in genetics. And I started my faculty position at the University of Pennsylvania School of Medicine in 2006, and I'm in the Department of Biostatistics, Epidemiology, Informatics. And since I'm a faculty in the School of Medicine, people know that I'm in the soft money-based position. In fact, 80% of my salary is coming from grants. So after I started my job at the University of Pennsylvania, GWAS got started. And I naturally started to work on GWAS myself and published many papers in this area. But I quickly see that this area was becoming very crowded. You have to have a lot of resource, be involved in large consortia in order to publish high impact papers. So at that time, I started to think what I should do next for my research. I remembered I had a conversation with Rafael a long time ago. At that time, we were both members of GCAT. And I asked Rafa, I wanted to make a decision. Should I go into RNA sequencing or should I go into epigenetics? So in the end, I didn't work on epigenetics. I decided to move into RNA sequencing. So I worked on problems related to RNA sequencing for a few years. And then naturally, around 2015, I started to work on single-cell RNA sequencing because the single-cell technologies just started, and I thought it's going to be a very promising technology, and eventually it's going to be like GWAS many years ago. So that is what I started doing in 2015, and I worked in this area for about 5 years. And then near the end of 2019, I started to realize that single-cell sequencing is becoming crowded again. Maybe it's the time for me to move on. And then I saw a number of papers, in Nature Methods and Nature Biotechnology, and I saw those interesting papers on spatial transcriptomics. And I thought maybe I should work in this area myself. So I started to read many papers in the area and finally started working on my first project in this area in 2020. And we got the first paper published in 2021. And in the same year, I also started to work on a few problems related to the single-cell multi-omics. But through my work in spatial transcriptomics, I realized that, in the spatial transcriptomics data, there is often a companion very high-resolution histology image that is associated with that. As a person who was trained in genetics and genomics, I had no experience working with image data at all. I didn't even know how to read in an image file. And I talked to my colleague, Takeshi Hara, and asked him, how do you read in an image file? And he told me how to do that. So that is how I got started in the imaging field. But since then, my group has devoted a lot of efforts trying to learn how to process and manipulate the imaging data and how to integrate the histology imaging data together with the spatial molecular data that are generated from spatial transcriptomics and the spatial multi-omics experiments. So now, after learning more in this imaging field, I realized that for my next stage of the career, I should focus more on precision pathology. Because in precision pathology, we're going to integrate all sorts of modalities together, including single cells, spatial omics, and digital pathology. And there are a lot of computational challenges that need to be resolved. So in 2022, I obtained a secondary faculty appointment in the Department of Pathology and Laboratory Medicine at Penn. So I also prepared a few slides that summarized the factors that drive my own research. Both of my parents are trained as doctors. Although I'm not a doctor myself, but I really wanted to make an impact on people's lives. And I really appreciate the fact that I'm a faculty member in the School of Medicine at Penn. And over my last 18 years working at Penn, I feel that my perspective of doing research is completely changed. So when I first started working at Penn, my way of thinking was like a traditional statistical geneticist. I wanted to work on large-scale population-based studies, and I even thought that Penn is not a good place for me because Penn doesn't have a lot of large-scale population-based studies. However, over the years, I started to appreciate that in Penn School of Medicine, there are a lot of very good researchers. And the strength here is translational research. So over the years, my research interest has been shifted from basic science to translational research. And I feel that I'm very lucky to have identified a lot of scientific collaborators that share common research interests with me. But in the meantime, they also have a complementary expertise from me. So here I list, this list is incomplete. I list a few people that have deeply interacted with in the last few years. And these people have really shaped how I consider science and how I do my research. So I worked with, I can see a lot of people. I worked with a cardiologist, ophthalmologist, nephrologist, neurologist, pathologist, cancer biologist, microbiologist, any type of ologist that you can think of since I joined the faculty here at Penn. And I think this is a very rich experience that really benefited myself. So for example, Murdoch Reddy, I have been working with him since 2006. At that time, both of us were junior assistant professors, and we started working together on GWAS, and we published the paper in the Lancet. It's a GWAS study on cardiometabolic disease. Although we started working on GWAS together, but over the years, we have grew together in terms of science and research. So over the years, we did the GWAS, and then we moved to RNA-seq, and then we moved to single-cell RNA-seq, and then we moved on to spatial transcriptomics. And now we are working on something that integrates digital pathology and spatial transcriptomics together for cardiometabolic disease studies. And at Penn, I also have the opportunities to interact with like Dr. Vet Stambolin, who is an ophthalmologist. I work in age-related macular degeneration. I have been working in this area since I was a graduate student. And I really benefited from having this long-term collaborative relationship with an ophthalmologist. And the other person that I really wanted to highlight is Dr. Kathleen Sastak. My work with her is different from the traditional type of collaborations. So my work with her is always, her lab does all of the bioinformatics computational analysis. She has a lot of people who are capable of doing that high-level analysis. But my interaction with her is always like she has completed all of the analysis. She wanted to show the results to me and ask me for opinion, the feedback. And she will tell me all of the computational challenges that they have encountered when analyzing the data. I feel that every time when I have a meeting with Dr. Kathleen Sastak, I learn something new. And my interactions with her was always very stimulating. And through interacting with her, it motivated me to develop a few methods, including music, which was designed to perform cell type deconvolution that integrates single-cell RNA sequencing data and bulk RNA sequencing data together. And now, given that I'm working in the area of spatial transcriptomics and pathology, I naturally have expanded my collaborators to the pathologists. So I have two very good pathologist collaborators, Dr. Elizabeth Firth and Michael Feldman, and I learned a lot from them. So every time when we see some results, we don't really know how to interpret. We have no idea if the results are making sense. That is the time for me to schedule a meeting with them, show them what we have got and what are the questions they want to ask. And I also need their opinion to confirm our results are valid and biologically meaningful. And in the last couple of years, I've also expanded my research area into cancer. As Rafael has said, cancer genomics is a very exciting research area that many open, interesting questions that need to be solved. So here's a list of the diseases that I have worked on in the past, age-related macular degeneration that I've been working on since 2004 and 2005 when I was a graduate student. And I worked extensively in cardiometabolic disease. And together with Dr. Murdoch Riley, we have had many multi-PI grants, starting from GWAS to single cells to RNA-seq and to spatial omics and digital pathology. And I also worked on Alzheimer's disease, kidney disease, and more recently on cancers, including breast cancer and lung cancer. So although these diseases they seem to be seemingly unrelated, but they all share something in common. And I think that the common thing about all of the diseases that I work on is that all of the diseases are essentially due to chronic inflammation. So knowledge in immunology is really the key when you conduct scientific research. And through seeing the connections of all of these diseases, my interest is I wanted to develop statistical methods and computational tools that can facilitate my biological collaborators for their biological discoveries in the real human disease-based studies. So here's what I wanted to do currently. So over the years, I have worked in many areas of research, starting from the traditional genetics and up to single cells and genomics. And more recently, I have a very strong passion in digital pathology and precision pathology. I really wanted to integrate all of my skills together and to achieve one single goal. So what I'm really excited about doing in the next few years is to integrate spatial omics data and digital pathology data together to generate a large amount of virtual spatial or molecular data in the tissue samples that are collected in the biobank. If we can achieve this, this means that we will be able to link the spatial molecular information together with the rich medical record information in the biobanks and also the genetics. So we can form a loop, and we can use this type of information to build better diagnostic and predictive models for disease. And if we can achieve this, we will also build a big tissue reference. Eventually, I hope we can build the tissue references for every important tissue type and organ type that are relevant for human disease. And this type of data is going to be useful in many aspects. So for example, in the clinic, we can use this data to provide diagnostic support for the doctors. And for the residents and the fellows, we can use the data we created to help their educational purposes and training. And of course, this data will provide a good resource for the researchers to develop new tools, new methods, and to enable novel biological discovery. So here's a few things that I wanted to share with the people here. I think in order to do research, you need to keep the passion of learning. And the genomics is a very fast developing field. It's mainly driven by technology. So for us, for the methods developers, myself, I always follow the technology development. You have seen that over the years, my work is closely following where the technology is, starting from GWAS to RNA-seq to single cells to spatial, etc. And also, in terms of doing research, my own style is that I always identify a scientific problem first, and after that, I will decide what tool will solve this problem the best. And I also feel that it's very important to be deeply involved in disease-based studies because this is going to motivate many interesting scientific problems. As I've said, there are many open, interesting computational challenges that are waiting to be resolved. And one last thing I wanted to share is that try to be unique and find a niche area. I remember around 2005, I was about to graduate, and my advisor, Gonzalo, gave me advice. He said, regardless of where you go, you wanted to make sure that you are unique. You are not replaceable. I still remember his advice until this day. I think that is a really good advice. So when I decide what problems to work on, I try to think if that is a unique problem, if I can identify my uniqueness, and if this is a niche area that I have. So that's all. Thank you.
Thank you so much, Mingyao, for your very well prepared, detailed, and insightful introduction to your journey. So it's time to move to our Q&A time. So we will start with some questions prepared for our two panelists first. So the first question, which is also the same question we got from the Q&A from an audience, is about this AI challenge. What are your opinions regarding whether the current AI can challenge statistical models in the genomics field? Compare with computer scientist training, what are the unique opportunities for people like us with statistics training in the genomics field? Maybe we can start with Rafa for this question.
Sure. So in our context, I don't see the AI, you could call it revolution, is as something that new. So when we do our work, we often need to compute an expectation of some outcome given predictors or covariates, whatever you want to call them. And for several decades now, there have been innovations that lets us be more flexible when we do that. For example, so I don't know what it is now, 40 years ago, random forest came along, and as a method that permitted the computation of that expectation to be much more flexible than what we could do until then on paper, which was mainly regression, where you can actually like compute the coefficients by hand. But that when computers come and we start using computers, then a bunch of possibilities open up. So things like random forest, and then all these other, I'm just using that as one example, but there's all these machine learning algorithms are ways in which instead of assuming a linear model, we are more flexible. That's, as I see it, that's all it is. Now there's a new innovation that's actually quite significant now, and it's that the GPU, the emergence of these faster chips, GPUs, and very, very smart algorithms for optimizing functions, taking advantage of parallelization. That now permits us to fit even more flexible models than we could before in a finite amount of time. But so what we've gotten is just one more tool in our toolbox to estimate these things we care about. But I don't see it as a challenge of what we're like. It's like a competition. to what we do. It's still, uncertainty still there, variability still there, that's not going away. Like that there's that we were estimating expectation, but the plus epsilon part isn't going away. That's part of nature. And understanding that and being able to interpret the estimates that you get appropriately, given what the epsilon is still what is the main thing we learn in statistics, and that's still going to be around. That's not going away. So I don't really see it as a either or. It's again, it's just one more tool that we should all learn. I think 20 years ago, I think we should have all been learning machine learning, and it took us a while, but now it's being taught in our programs. And today we should be learning all these new techniques for estimating, for modeling very complex multidimensional curves. Is that like, I don't know if there's a follow-up question to that, but that's how I view it.
Thank you, Rafael. I think that's also my view of it. Yeah, thank you for your input, Mingyao. And
yeah, my view is similar. Actually, I do not think AI is a challenge or threat to statistics. And to me, when I conduct my research, I identify the problem first. And then I think about what tool solve the problem the best. It could be a statistical approach, or it could be a machine learning AI-based approach. It doesn't really matter. So I do not really make the distinction of, I'm a statistician by training, I should only use a statistical tool. I do not think about the work in that way. So I always think which tool solves my problem the best. In fact, in my work, I used both statistical approaches and also machine learning approaches. It just depends on the nature of the question I ask. And I think statisticians have a lot of strength in terms of doing machine learning-based work due to the rigor of our training and our ability to quantify uncertainties.
Thank you, Mia and Rafa. So as a follow-up question I have, actually, this is also from my conversation with some other people. That is, if we just want to solve the problem, there could be multiple ways. You could go with a more flexible model and try to regularize it until you have something satisfactory. Or you could start from more about like, okay, my understanding of the data, what are reasonable assumptions, and build a statistical model. Maybe in terms of the performance, ultimately they will be similar, but the developmental process may be more based on human understanding, intuition on the statistical modeling side, or computation heavy if you go with a complex model first and then shrink it down. So which way would you think is more desirable, or you think it's just a personal choice based on the researcher's expertise? Maybe
Who do you want to go first?
Yeah,
Me?
Rafa, maybe you can go first. Yeah.
Okay. Yeah, I think the understanding what's going on approach is better always, if it's possible. If you're trying to predict an offspring's height from their parent, the data is bivariate normal, so a regression model is going to work, and the coefficient is going to be super interpretable, right? It's like how much of the height you inherit from your parents. That's what I'm going to use. But if I fit a deep learning model, it's gonna give me back a line. In fact, it might be a little bit better because maybe the edges are a little flat. But in terms of what I gain from the first and the second, the first one obviously gives me much more. So if I can come up with a method that connects to something interpretable, of course I'm gonna choose that. But I'm not gonna limit myself to using only those models, especially in cases where prediction is all that matters. That's not as common in basic science, and maybe even in cancer, it might not. If you can actually predict if someone has cancer or not, and you don't know how you do it, it's still, if it works and you can show it works, it's still a great thing. So I'm not going to limit myself to using only interpretable models because I can't interpret them, because the reality is that usually biology is too complicated for simple models, and in fact something more black boxy might work better at the end of the day. But just to echo what Mingyao was saying earlier, you shouldn't be constrained in any way. You have a toolbox, you think deeply about the question, and you pick whatever tool will best help you to answer the question.
Thank you, Rafa. Mingyao, your input.
Yeah, so regarding the question you asked, from my own experience, I never had that struggle. Because when I work on a problem, I have to think about, I rely on my intuition, my intuitive understanding of the problem from the biological perspective. So for example, when I worked on cell type deconvolution, I have to think about, the bulk tissue, and it's composed of cells from different cell types. And we have to consider many other factors, including the cell size, etc. So those rely on your biological knowledge. So these drive how I derive the model. Yeah, I never encountered the struggle that other people have.
Thank you, Mingyao, for your input. Thanks, Rafa, also. So as a follow-up question, which I think is everybody faces in a popular field, and Mingyao mentioned this briefly in your leadership journey, which is that as a topic becomes popular, there will be numerous methods developed in the field, right? But we didn't do data generation. We're not experimentalists, so we don't own the data, or we are not biologists who knows the problem the best. Then how can we play the leadership? So how can we let people know, okay, we are driving the research instead of just playing a supportive role? Maybe Mingyao, you can answer this question.
Sure, I'm very happy to share my experience. In fact, I think the benefit of working in genomics is that there's a lot of publicly available data. Yeah, looking at my own methods work, most of the times I do not rely on my collaborators generating the data for me because they are too slow. So what I do is that I read the top tier journals like Nature, Nature Biotech, Nature Methods, etc., and try to anticipate what is the next wave of technology development, what will be the most popular technology that researchers are going to utilize. And then I utilize the public available data in those papers to develop my methods. So this way I can be ahead of my collaborators. And when they see my papers, they will know I'm an expert in this area, and they will ask me for advice. So this way I have a leadership role in the project. So oftentimes I'm a multi-PI on a disease-based study with my collaborator being the contact PI. So that is my own experience.
Thank you, Mingyao. What's your input, Rafa?
So I've been very lucky in having really good collaborators, but I don't necessarily try to predict what the next big thing's going to be. I have people come to me with questions and looking for help, and that has led in the past to me working on technologies that are taking off, and then a bunch of people start using. But I had early access to the data and early access to the challenge. So and so that's in that sense, I do let the biologists or my collaborators lead the biology part of the project. And what I've done is in my career, and this is very personal, so I'm just answering the question from my perspective. We then realize that whatever solution we came up to, it doesn't always happen. In fact, I would say it happens less often than not, that the solution we came up with is something that will be more widely applicable. Maybe because two things can happen. Either many people are going to start using this technology, and this very specific algorithm we developed for it is going to work well for that. And then you write a paper describing it, you get a lot of citations, you write software, you get a lot of downloads. But another thing that happens is that you realize that you came up with an idea that actually applies across many different genomics technology or even beyond, right? So then that's like the story of applied statistics. So we work on that. So I do let my collaborators take the lead on what they're experts in, and I focus on what I'm good at, which is methods and software for the analysis of data.
Thank you, Rafa. Yeah, so I think both you and Mingyao mentioned two kind of, I would say, niches in statistical genomics. One is that we can try to solve the scientific problems or have something diagnostic, translational ourselves, or we focus on the methodology and try to develop something general enough. Thank you. Yeah, so another follow-up question, which I think is more practical in the sense of how to help our trainees find academic jobs if they're interested in academia, right? So I think we face this challenge in this interdisciplinary field about where to publish, right? Our students might have opportunities in the biostat department or in the cancer center medical school department. But maybe the two different places are having people in the review committee, hiring committee looking for different journals or the channels to publish. So for us, we know that if we publish in science journals, we may get more impact. However, if our students are looking for jobs in the biostat department, then many of the colleagues there may not be aware of these journals or familiar with the journals if they're not in our field. So how would you address this challenge for your trainees? Maybe we can start with Rafa. You have many trainees already.
No, that is a challenge. So the first question that I ask each one of my postdocs, and graduate students don't know early on, you have to ask them later. But definitely postdocs, what do they want? So by the time they come to do a postdoc, they should know if they want to go to industry, they want to go to a biostat department, or they want to go to some other much more applied type of department, comp bio type, systems biology, those types. So if they want to go to a biostat department, you basically, your question describes the challenge. And I don't know if I have the perfect solution, and it's unfortunate that we have to be thinking about these things, but it is better. My experience is that if you're more of a generalist, it tends to be better. You seem to have better chances. The exception to that is when it's an area that's it's so fashionable and hot that the deans are putting pressure on the biostat department, or there's some money pot to hire someone in like single cell. My most recent one like that was somebody needed a person in spatial transcriptomics. That's what they wanted to hire, and it just turns out that one of my PhD students was working in that area, so they actually got that job. So both things happen, but it's much, if as a general answer to that question, if you want to go to a biostat department, I try to train my students to be more general, to try to whatever method they develop, try to make it as abstract as possible, so that they can get publications in statistical journals that all of the faculty in the biostat department will recognize. And I don't know if that's the best for science in our discipline, but that's because I know that's what's going to work best for them. That's how I steer them. So that's my answer to that question. Yeah, if their answer is I want to be faculty in a biostat department, I will steer them a little bit towards working on more, trying to make their method more abstract as possible. But of course, you don't want to go too far and in a way that's unnatural. That's not going to work, and it's definitely not worth it. Like do a bunch of derivations you don't need to do and anything like that. But there's ways to do it. You can write an algorithm and put it up on that uses statistics and put it up on Bioconductor or GitHub, and people start using it, and you get a nice paper that gets cited a lot, maybe even in one of the glossy journals. But then when a biostat faculty is reviewing that, they don't know what to do with it. Like they read the glossy journal and they don't understand really what you did, because it doesn't really explain it, because it's focusing on other things, and then the code is hard to. So you can take the time to write that up, write it down so the statistician can understand it, focusing on the statistical thoughts that were key to the thing, to whatever you did working, and publish it in one of these more well-known biostatistics or statistics journals. With that said, I'll say one more thing, and then I'll let Mingyao answer. There are some journals now that are publishing that let you put in quite a bit of details for the statistics. Like Genome Biology is my favorite in that regard. They do let you write statistics papers, and it has a much wider audience than any of the stat journals. So then when a statistician that isn't in your field reads it, they'll still understand some of it because you're writing down a model, you're writing out derivations, you're writing down estimates and confidence and how you estimate standard errors, all that's in there. So that's my answer to that question. It's a hard one, and I don't know if I'm going to be in the same place in 5 years with my answer.
Thank you, Rafa. Yeah, Mingyao.
Yeah, I can share my personal experience. Yeah. So in 2005, I was looking for jobs. I interviewed at a few places, and there was one place they said your area is too specialized. So in the end, I didn't get a job offer. But luckily, at that time, Penn was specifically looking for a person who has expertise in statistical genetics. And because of that, I got the position here, and I stayed here since then. So there is a plus and minus in terms of the direction you choose. I don't really know which one is the best. I think my suggestion is just follow your heart. Which one is of your interest the most?
Thank you, Mia. I agree. Yeah, that's always right. Follow your heart, follow your passion. Yeah, that's why we do this. So now I will leave the remaining time for the questions asked by our audience. Okay, this is a general question. What's the next revolutionary technology? Maybe Mia, because you said technology is the driver, right?
The genomics technology?
Genomics, yeah, I think we should just focus on genomics, yeah.
I think, since I work in spatial, I feel that eventually everything is going to be spatial omics, all sorts of spatial omics, and also imaging. I think imaging is another area that we should pay attention to. That is my personal opinion.
How about Rafa?
Yeah, so I don't know how much in the future you want me to extrapolate, and it's like Yogi Berra said, predicting is hard, especially the future. So I'll predict the present. So current, like Mingyao mentioned, right now, you can see some technologies that are starting to come up, and it looks like they might be widely used and have the kinds of properties that make statistical thinking important. Spatial transcriptomics is definitely one. That's not that. I mean, that's been around for quite a little bit, but it's getting more and more. Precise in how small, how the resolution is getting better and better, and that changes the problem. But it's another one that I'm starting to see more and more is in cancer is early detection via something called cell-free circulating DNA. That's not new, but it's so hard that I haven't really seen any good solutions for that. There are some companies starting up out there that looking at either DNA or methylation, but that's a very hard problem where the key challenge is that the signal to noise ratio is almost zero. So it seems like a very good problem for statisticians because of that. You really have to get that estimate to be as precise as possible because it's accurate as possible because it's again very low signal to noise ratio. There's all these other things. Molecular imaging is also becoming more popular, as pointed out, and also there's now long read DNAs that will maybe change the way we quantify RNA transcription, but also other things like methylation. That these things can measure methylation in a very interesting way. It has to do with how the intensities look. If they look a little bit off, it's because the C is methylated, so it's like a signal processing challenge. So yeah, those are some of the things I see, but it is that the answer to that question, it's very hard to predict more than like, I don't know, 6 months ahead of time, but it's also changing so fast that yeah, it's the I guess the question I'm answering a different question, which is right now, what are some of the technologies that look like are going to get a lot of attention? And I'm not sure yet, but that's what I'm guessing based on how many people are coming to me with questions with those technologies. The other general area I would say is immunology in general is also getting a lot of attention in cancer. mainly there's many reasons, but the main one is that they've come up with these ideas where you can trick your body into your immune system into actually killing cancer. And that the success of some cases in that area has made immunology come to the forefront in cancer. So that there's a lot of different ways you can measure this, and and different kinds of technologies that are being used to understand the immune response to cancer and the behavior of the immune system around cancer cells. There's a lot of opportunities for people who are, trained as data analysts.
Thank you, Rafa. Yeah, thank you, Mingyao and Rafa, for your very insightful answers. The next question was asked by two people. The question is, have you arrived at any best practices for collaborating productively with domain experts? Maybe, Mingyao, you are in a very good position to answer this question.
Wait, I'm sorry, what did I missed? What did you say?
The best practices for collaboration.
Oh, for collaboration.
Yeah, I think my experience is that you have to learn the domain knowledge yourself. In the very beginning, it can be hard. I remember when I started my job at Penn in 2006, a few people came to me, wanted to write an R1 together. So one of the proposals was on heart failure. I had a hard time to understand what it is about, because there are a lot of jargons, new terminologies, and disease-specific materials that I do not understand. But don't be frustrated by that, just to keep learning. Over the years, you should become like a domain expert in a specific disease area. So for me, I have been working on cardiometabolic disease and heart-related disease since 2006. And now I can very easily communicate with any cardiologist. And if you are deeply involved in disease-based studies, just keep accumulating your knowledge, and eventually you will be, you have the freedom to speak using their language. And this is how you can best collaborate with the domain experts.
Thank you, Mingyao. And Rafa?
Yeah, so collaboration, I would say that my main advice, based on my experience, is to choose carefully. You want people who, so rehab, the demand for our expertise is much higher than the offer, so we do actually get to choose. So knowing that, you want to avoid people who are not respectful of statistical thinking. So it's okay. I have worked with people who have no idea about statistics, but they are actually aware of that and don't try to override your advice because they don't know. That's okay. It's just that there are some that think they know more than they do, and it's just not good to work with people like that, because you end up, your advice isn't necessarily always listened to, especially if you get the conclusions they don't want, and that's terrible, because then you might even they might end up writing a paper that's wrong, and they add you as an author. It could be bad, right? So you want to avoid that. And that's becoming, it used to be harder to find people like that. It's becoming easier now. Our field has gotten a lot more respect in the last 10, 15 years. I think in part thanks to the success of our discipline in genomics. So I would say that's one. Another one that I've had challenges with is and this is a little bit it's a little bit tough on the people that are starting up and still are trying to figure things out, but sometimes a collaborator comes to you and they don't actually know what they want to know. They don't have a question. And that's hard. I mean, you could maybe you can argue that it's part of our job to teach them to ask questions, but it seems like sometimes it seems like it's too much because we're not experts in their area. But that rarely leads to a good collaboration when they come to you basically saying, just find something. I just ran all these measurement technologies on whatever I'm interested in, and I just go find something. Look at this paper. I want a paper like this one. So you want them to have the more specific they can be on what they want, the better. It makes everything smoother and more productive. The reason I'm a little bit hesitant to give that advice. Is that's gonna be a little bit unfair against early career biologists that are still trying to figure things out. But if you're an early career statistician yourself, then it's a little bit unfair for you to be doing to spending so much energy and time just getting going with the project. Just even figuring out what are they asking. So that's the second thing. And the third thing I would say is when it comes to data analysis, trust yourselves. One thing that happened to me very early on is especially when I first got into the field, I would go to talks by computational people and I wouldn't understand a thing they were saying, and nothing made sense. And I thought I was just not trained yet. But they were talking about data analysis. They were talking about take data, and apply math to it and come up with an interpretation. Later in my career, as I got more experience, I realized they were just full of BS. They were just not. There was a lot of those back in the 90s. So trust yourself. If you don't understand something that relates to your area of expertise, chances are it's not you, it's the collaborator that's not really understanding something that you do. So that would have saved me a lot of time back in 1999 if I had been more confident about statistical thinking being one of the biggest keys to the analysis of genomics data.
Thank you, Rafa. Thank you, Mingyao. Yeah, so as we are running out of time, maybe we will use the last question to answer. There are two people asked the same question about advice for junior researchers. How can they find good collaborators? Because maybe unlike you, Rafa, junior researchers wouldn't have many people coming to them. Then how would they reach out to good collaborators?
Well, I think that's then, the chair of your department isn't doing their job if you're not getting people approaching you because there's so much demand. So it's not that easy to go find people as it is just when people come to you, because often if you're in especially if you're chasing like famous labs and famous PIs, my experience in this field is that really famous PIs that are actually in the lead of these great technologies and they're part of these big consortiums in NIH, they already have their team of data analysts. They're usually terrible. That's why you see all these crazy papers in the glossy journals when you read it and you can't believe that this has been published with the statistics being so terrible. That's because some famous PI hired some undergrad and that knows Python and they did all the data analysis. That's why that happens. If you're wondering why you just see all this, they're not going to want to work with you because they're control freaks and they don't want some other PI involved. So that's not necessarily the kind of collaboration it's not. That's why it's not always good to go out and find people, although you definitely can. And I have found people that way, especially when one of my students is interested in a particular area. It does work, but it's just don't if your chair is doing his or her job, you should be getting, and you're working in in a cancer center or in a biostat department, you should be getting, if you're looking for collaborations, you should be getting them. Now send me an email if you're not. I'll talk to your chair.
Thank you, Rafa. How about Mingyao?
Yeah, my advice is, when you start your job, try to give as many talks as you can. This is the best way to advertise yourself. Yeah.
That's very straightforward. Thank you. Yeah, and maybe one last question. I think because this is very important and we can wrap up from here. The question is about the rapid technology development, which will give us very little time to evaluate the robustness of each method, or sometimes didn't give us time to think deeply, because if we do, maybe we are behind the curve. Then what would be your suggestion on this challenge? Yeah,
I don't really know. I struggle with this challenge myself. I feel that the field is moving so fast. When I was a graduate student, the field was moving very slowly. So I can spend a lot of time reading one single paper. I cannot afford doing that anymore. I don't really know. I don't have a good answer for that.
I would say, well, my experience is that it's okay to do things right. You don't have to publish in the glossy journals to be successful in our field. Yeah, so okay, so there is, we are famous statisticians in this genomics world for being too slow. So you don't you want you don't want to publish a paper today about how to analyze microarrays. That's then too much time has gone by. Now they're not that interesting. But at the same time, I would not be discouraged by the fact that you didn't get you weren't fast enough to publish the nature paper, science paper. It's okay 2 years, 3 years later that you publish Lima in, like, I don't even know what journal it is. Statistical Applications of Genomics and Molecular Biology, I think I don't know if it still exists. Lima has, I don't know, 30,000 citations. That paper was published in I don't know if you all know what Lima is, but it's probably the most used statistical idea in genomics. Maybe that or the Q value. I don't know. It's one of those. It got published in 2004. Microarrays come out in like 1995, 96. That's like, that's 8 years after the thing comes out. That's fun. It's super successful, and actually it is used with other technologies now. That's just one example. I can give you many others, but it's okay to wait a little bit and do it right. But it's true that balance is, I guess you learn it with experience. Can't take too long to then wait until the thing is completely obsolete. Yeah, but I think we should all be okay with not being the first to put out some like glossy journal paper.
Thank you so much, Rafa and Mingyao. This is a very insightful discussion. To wrap up, and also this is motivated by a question we didn't get to answer, is about the history of statistics, medicine, biology. I wonder whether you can recommend maybe 1 or 2 papers from the history that you think people should read if they want to find the passion for statistical genomics, or the paper you personally like.
I have to say I don't have a paper
Or
to
book.
recommend because
Yeah.
the field is moving so fast. I cannot identify one single paper that impacts the way I conduct my research. Maybe Rafa has a better answer for this.
Yeah, it would be unfair to just name 1 or 2, but
Yeah.
you know what? I think if you're working in medicine in general, there's these Altman papers that were written, I think, in maybe the 70s or 80s, and they're really good, and they're still relevant today.
So how do you spell it?
ALTAM. He's a very famous statistician.
ALTAM. Okay.
Oh, Altman.
Yeah.
Yeah.
Those are
The fact at Penn State, right?
I think so, but it was like the whole series. It was a whole series.
Yes, in Nature Methods or Nature, there's a whole series of short articles.
Yeah, I also read them. Yeah, these are good.
No, I think I'm talking about something different.
Okay.
I'll put it in the chat if I find them.
Okay.
I wasn't ready for this question. Sorry.
No. Yeah, thank you. It was just a spontaneous question. But Mingyao, I think I read the thing you talk about, but for Rafa, he might be talking about something else. Yeah, but I just think it's very important to be unique, right? Something useful for us may not be the same useful for other people, but think about the questions they're passionate about is most important. Yeah. Thank you so much, Mingyao and Rafa. I think it's a very informative session for me and for our audience, and your sharing of your experience has been very helpful for our field. Thank you for your time and participation.
Okay.
Thank you so much for the invite.
Thank you.
Yeah, same here. It's Doug Altman is the name.
Sorry,
right.
how do you
Doug, D-O-G, Douglas Altman.
Douglas Altman.
Okay.
All right.
Okay. Thank you. Thank you,
Thank
Mingyao.
you. Bye-bye.
Bye. Thank you, everybody.
Bye-bye.